non-invasive brain stimulation. Please take it away, Molly.
Thank you. Thank you for inviting me, and thank you for organizing what so far has been a totally awesome workshop. I'm going to talk about many different things today, but I'm going to focus in the end, the sort of empirical meat is going to be related to understanding neural entrainment using non-invasive brain stimulation. So I don't need to set up too hard with you as the audience, but the reason that many of us, all of us maybe are here is because we're interested in the fact that humans in particular synchronize our bodies with each other and with sounds when we're dancing, when we're walking side by side, when we're making music together, when we're engaging in sports together, and when we perform rituals. And this synchronization with each other is thought to underlie bond forming and maybe social cohesion. But my perspective is that we can't consider how we achieve synchrony with our bodies and with each other without considering that we have brains embodied in these bodies that we're syncing with, that we're synchronizing with. So the starting point for all of the research in my group is that brain rhythms synchronize with the rhythms of sounds. This is a waveform of speech, so this is a sentence that unfolds over time, and you can see that speech has fluctuations in loudness that give it a rhythm. And we know that brain rhythms, this is a cartoon of rhythmic brain activity, we know that brain rhythms synchronize with rhythms in sounds like these. By now, there's a decade or so of research showing that successful synchronization between the brain and the environment is associated with better comprehension for what's been said, better memory for what's been said, and also better success at solving the so-called cocktail party problem. So it's separating, paying attention to one person's voice in a noisy background. So by now, the field seems to have arrived at a consensus that brain-environment synchrony is good. And the explanation for why this is true is rooted in understanding rhythmic brain activity as reflecting fluctuations in neural excitability. So I'm not going to get too deep into any complicated neural analysis, but the idea here is that any complex full-band brain signal that we measure from cortex can be analytically decomposed into narrow-band brain rhythms. And we typically give these brain rhythms labels. These are the canonical frequency bands that we talk about, that we read about. This is a cartoon, let's say, of delta activity, slow activity. And the idea is that instead of having a meaningful y-axis like voltage, for example, we can put excitability on the y-axis. So what that means is that these fluctuations are fluctuations between up and down states, states when the bit of cortex where this rhythm is being generated is more excitable. That means the neurons in that region are more likely to respond to sensory information from the environment. So from the perspective of a single neuron, a neuron that lives in this sort of cortical neighborhood is going to be more likely to respond to sensory input at some times at the more excitable phases of this oscillation than others. So from the perspective of that single neuron, we can operationally define excitable phases and inhibitory phases. And it's maybe important to note here that our understanding of these excitability fluctuations comes not in the context of entrainment originally, but from measuring ongoing endogenous oscillations. So those are oscillations that are just happening when we're not doing anything or maybe not engaged in a task. So this is brain activity that's just happening. And when we talk about entrainment and the usefulness of entrainment, we make a little bit of a leap here. So our assumption is that since excitability shapes the timing of neural firing on its own, if we could align our brain rhythms with the rhythms in a sound, that would align these excitability fluctuations with the rhythmic structure of the sound, meaning that neurons are going to be more likely to fire at some time points than at others. And when a sound is rhythmic, synchrony does a good job of lining up the brain's excitable moments with moments in the sound where important content is likely to happen. So this is a mechanism for dynamic attending. We can attend, at times that we need to, but we can sort of take a rest at times where we don't need to be attending. And so this is thought to be, proposed to be, a metabolically inexpensive way to selectively attend in time. But this depends on our assumption that those endogenous rhythms that reflect excitability, that we know reflect excitability fluctuations, can become synchronized with the sound. That's a little animation mistake. So how do we typically test whether entrainment is happening? This is a stimulus rhythm. This is an amplitude modulated sound. It fluctuates in loudness every 300, it has a peak every 333 milliseconds. So this is a 3 hertz oscillation in the stimulus. And we can transform that stimulus into the frequency domain. So now we have frequency on the x-axis, amplitude on the y-axis, and you can see a peak at 3 hertz that corresponds to that 333 millisecond fluctuation in the sound. These are EEG data that were recorded during listening to that exact sound, and we typically transform those data also into the frequency domain, and we look for the presence of a 3 hertz peak that corresponds to the rhythmic structure of the stimulus. And this shows us that brain activity is synchronized to that stimulus. So what this means is that if we were to filter the brain activity so that we were only looking at 3 hertz activity, we'd find that it would be aligned with the temporal structure of the rhythm. And there are many different ways to do this. I'm talking about transformation to the frequency domain. You can transform to the time frequency domain. You can also use a narrowband filter and calculate something like coherence, for example. These are all methods that lead us to the same conclusion, that brain activity is synchronized with sound rhythms. And when we observe that synchrony, we usually label it entrainment. So one question the field has started wanting an answer to is this synchronized brain activity truly entrained? So we know based on our analysis that it's synchronized to the rhythm, so what's the difference? What do we mean when we say that a brain rhythm, or any rhythm, is entrained? And your answer to that question depends a lot on your definition of entrainment. I'm going to be pretty strict during this talk, but I don't necessarily apply that strictness when I'm reading papers or discussing with you, for example, but for the sake of thoroughness, I guess, I'm going to give you my strict perspective. So what we mean is that rhythmic brain activity, rhythmic neural activity, is generated by an oscillator. And that means that rhythmic brain activity is governed by the same mathematical and physical principles as any oscillator, like a pendulum or our circadian rhythm, for example. For a neural oscillator, we tend to adopt a certain set of criteria. Not that I don't have beef with these criteria, but I'm going to come back to that later. So these criteria tend to be these. First, the oscillation has to exist on its own. It's not created by the stimulus. It exists already, and it just becomes synchronized with the rhythm. In the presence of a stimulus, so this is what we mean when we say ongoing or endogenous activity. This is, like I said, the activity that's just going on. In the presence of a stimulus, the oscillation has to adjust. This isn't arguable, this is definitional for entrainment. The oscillation has to adjust to the stimulus rhythm. So any two pre-existing rhythms that have the same period, that have the same frequency, will be synchronized with each other, but that's arbitrary, right? There has to be some adjustment of the adaptable system to the environment. Then, once the stimulus is gone, the oscillation continues. This is the self-sustained property of oscillators, and we'll come back to that. So why does this distinction matter? Why does it matter whether a synchronized brain activity is really entrained or not? This is actually a mechanistic question with implications for attention and for cognition. So here's an example stimulus rhythm. One option, the entrainment option, is that an endogenous oscillation becomes entrained by that stimulus rhythm. But there's a second option, which we would call evoked activity, which we also know exists in the brain and is a huge contribution to our MEG and EEG signals. The biggest contribution, I would say. So here the idea is that a stimulus event happens and the brain responds. Another stimulus happens and the brain responds, and so on. So these are like event-related potentials, or ERPs, that we typically analyze in EEG data. The difference between these two options is that entrainment is predictive and evoked activity is reactive. So what do I mean? I mean that because entrained oscillations are in principle self-sustaining, they can go on when the stimulus stops or when you can't hear it because it's being masked by another environmental sound, for example. So when a future event occurs that's in line with the rhythm that we have been listening to, an entrained oscillation will have us ready for it. But the evoked activity won't, because it's not being driven by the stimulus anymore. It's not being created by the stimulus, so it's going to cease to exist when the stimulus ceases. So we need this predictive sort of function for things like dynamic attending. And evoked activity by itself isn't going to support that. So from a cognitive perspective, it does make sense to try to tease these options apart. So how can we do that? How can we tell the difference? I've already showed you that a common signal processing technique is to transform a time domain signal into the frequency domain, or something related. So if I transform this entrained oscillation to the frequency domain, we look for the corresponding peak in the frequency domain, and we say that the oscillation is synchronized to the stimulus rhythm. Here, the stimulus rhythm arbitrarily has a rate of 1 hertz. The problem is that with evoked activity, since it's being driven by the same stimulus, we also will still have that same peak, because something's happening periodically. Here, every 1 second, it's just a different something. So you might pick up on the fact that the harmonic series in these two frequency domain representations look different. That's not helpful, unfortunately. The only time that we'll ever have this single peak situation is in the case of a pure sinusoid, and that's never what brain activity actually looks like. This inability to rely on the harmonic series in a frequency domain representation is especially problematic when the stimulus gets even a little more interesting than an isochronous pulse train. So now imagine alternating between strong and weak beats that give rise to some kind of hierarchical structure, like a very simple meter. So this would be back and forth between strong, weak, strong, weak. And if we imagine then that we have nested oscillations, so 2 oscillations at the same time, one entrains to the sort of subdivision and the other entrains at the meter or the emphasis rate, we're already introducing harmonics into our assumed oscillation. So signal processing alone is not going to save us when we're trying to separate entrained from evoked activity. So how can we tell the difference between entrained and evoked activity? The simple answer to this question is we can't. At least not in human MEG, EEG data. It's not possible. So what can we do? It's not dire, I don't think. Can be frustrating, but I think the answer is in integrating across converging methods, and there's no one study that's going to by itself solve this issue. We're not going to talk about it, but there have been some recent modeling efforts that are quite good along these lines. Non-human animal data can actually be quite informative here, and we can get a long way with creative experimental designs that incorporate human behavior. And I'm going to spend most of my time talking about non-invasive brain stimulation today. And I will, at the end, emphasize how important it is, since oscillator models make very specific predictions. We can mathematically sort of derive predictions that we focus on theory and we derive testable hypotheses for ourselves. So I'll spend the rest of my time talking about these. I'm going to start by talking about the informativeness of non-human animal work for this dilemma. So I'm going to talk about macaque work using specifically recordings that penetrate the cortex. So this is an electrode, and it has those little black dots are contacts. So those are recording sites on this electrode. And this is cortical depth on the y-axis from superficial, the surface of the cortex, to deep, so inside. And cortex has different layers, and we can group them into categories. So we have supragranular, granular, and infragranular layers in the cortex. And the reason that these are different from each other is that these cortical layers have different types of neurons, and they also have different connectivity patterns to each other and to different parts of the brain. So for example, some layers, the deeper layers, are more likely to respond to stimuli from the environment, and so contain evoked responses. But other layers, like the supragranular layers, for example, might have more what some people say is computational functions. It's hard to say exactly what that means, but basically more top-down drive in these supragranular layers. So this is a plot of current source density, and I'm going to give you a super quick primer about the different signals that we can measure in an experiment like this. So current source density is a lot like what we measure in MEG and EEG. It contains slow activity, that's because of the way that it's filtered. So oscillations, if they're present, and evoked responses, if they're present. It can be measured in supergranular, granular, and infragranular layers. And the important part is that slow oscillatory activity is present in supergranular layers in the absence of a stimulus. So we measure endogenous ongoing oscillations in the supergranular layers. There's another signal that we can measure, which is called multi-unit activity. Multi-unit activity really is thought to be a quantification of neuronal spiking. So this is evoked activity. These are neurons that are responding to stimuli in the world. Multi-unit activity is measured in granular and infragranular layers, so these deeper input layers. And neuronal firing is only present in response to stimulation. Other than spontaneous background firing, we know that's present, but this kind of organized firing where you have a pattern of not firing, the dark red is where there's lots of neuronal firing, and the yellow is where there's very little neuronal firing. That kind of organized pattern of firing you only have in the presence of stimulation. So the paradigm in this experiment that I'm going to tell you about is to listen to two rhythms at the same time and to pay attention to one of those streams. So that sounds like this. Okay, and the monkeys are paying attention to one of these streams. We know that because there are infrequent targets in each stream, and the monkeys have to indicate when they heard a target in one of the streams and ignore the targets in the irrelevant stream, so you know that they're selectively attending. You probably could hear that there's a big frequency difference between these two rhythms. So that was, in this case, a 2-octave frequency difference, and that's going to be important in just a second, but let me orient you to this plot in terms of the paradigm. So now I'm plotting two tone onsets on our plot. I've just taken a sort of, a snapshot of the sequence. And you can see that the neural responses are structured and reflect the rhythmic properties of the stimulus. So I'm going to zoom in here on one bit of the supergranular activity, and you can see it's alternating between positive and negative. So that's an oscillation that's entrained by, well, we would say entrained by, I'll show you evidence for that in a second, by our stimulus. And the strength of neuronal firing, which I've already shown you, is also coupled to those fluctuations, as we would expect if, first of all, evoked activity is being driven by the stimulus, but second of all, if evoked activity is being organized by fluctuations in excitability that are represented in this supergranular slice. Here there are tones evoking activity, so we can't tell the difference between those two options. So here's the trick. Auditory cortex is tonotopically organized, so that means that any specific neuron or group of neurons will like and respond to one tone frequency, but won't like a frequency that's far away from that. And that's organized spatially in primary auditory cortex. So any neurons we record from are going to have a best frequency. They're not going to like frequencies that are 2 octaves away from that. So we can record from neurons who really like the stimulus frequency of one of those rhythmic streams, and we can record from another group of neurons that don't like that stimulus frequency. So when we record from neurons that like that frequency, this is multi-unit activity. So unsurprisingly, they fire a lot when a tone is presented. That's their favorite frequency. This makes a lot of sense. And if I plot the current source density, we can see what looks very much like an oscillation with evoked activity added to that. And we can't separate here in this signal between the two. When we record from neurons, different neurons, that don't like that same frequency though, they don't fire. They don't like that frequency. However, current source density fluctuates in exactly the same way. So we see slow oscillatory activity from supergranular layers is entraining to the stimulus sequence, even though that sequence is not evoking responses in the neurons in this area. So invasive recordings like this allow us to see entrained oscillations even when there's no evoked response. So I'm going to switch now and talk a little bit about behavioral work. I'm going back to our speech example. So again, the idea is that excitability fluctuations become coupled to the rhythmic structure of sound. That means neurons are more likely to fire when important stuff should be happening. I'm an experimental psychologist, so I don't know when neurons are firing or not firing. I don't have access to that information, so I wanted to approach this question behaviorally and provide another angle onto this distinction. So the idea that we had, and others of course also had, is that if we could sort of zoom in on these neural excitability fluctuations, and if we could present near threshold targets at different phases of that neural excitability fluctuation. So what I mean by near threshold targets are little sounds that you hear 50-50. Half the time you hear them, half the time you don't. And so we suspected that those times when you hear them are more likely to be when that bit of cortex is excited, and the times when you don't hear them are more likely to be when that bit of cortex is inhibited. So we thought if we could sort of probe all the way around the neural excitability fluctuation, we'd be able to see a pattern of target detection performance that mimicked that. So in high excitability phases, our brains are more likely to respond to, we're more likely to perceive and become conscious of targets. This behavioral prediction is not made by an account based on evoked activity. It has been pointed out that maybe it could be, but maybe a lot of things could be. This falls out really nicely from a neural excitability perspective, but it's a tougher one to derive from an account based only on evoked activity. So here's how we did that experiment. This is our sound. This is a constant amplitude sound, and you can see that there are little gaps here. Those are our targets. They're just little silences that we leave out of the sound. The participant's job is to detect those little silences, and we adjust them individually for each person, so they're right at 50-50 for you, duration-wise. The rhythm here is communicated by fluctuations in frequency, and that's important because the rhythm has a constant amplitude. It's on the same amount all the time. So we can put those little silences everywhere, and it's fair in terms of detection. What you can see, let me play that for you real quick. And there was one of the little silences. I made it really big so you could hear it. They were never that big. So what you can see here is that we have gaps at two different phases of the stimulus rhythm. So one kind of in the trough here, one kind of in the rising phase here. And over the course of the whole experiment, we presented many gaps at many phases that were spaced around the cycle of the stimulus rhythm. And the idea was that if brain rhythms are synchronized to the stimulus rhythm, we're also probing detection performance at different phases of the entrained neural oscillation. So first, we have to show that the brain was actually synchronized to the sound. I'll do that in exactly the way I've already described. So this is a frequency domain representation of EEG data that were recorded during this task. And you can see that there's a peak here. In this experiment, the stimulation rate was 3 Hertz. So we know that brain activity of some sort is synchronized to the sound. And now I'm going to show you behavioral data for the gap detection task. This is just for a single subject. So on the x-axis here, we have stimulus phase. The black thing is a representation of the stimulus. So here gaps would have fallen in the peak, here gaps would have fallen into the trough. We can also plot brain phase here. So we can do an analysis where we look for each gap, what was the brain doing right before that gap happened? And we can sort every single gap into a sort of phase bin and plot the data that way. It looks the same. So we can plot this in terms of stimulus or brain phase on the x-axis. And hit rates on the y-axis. So what you can see is this person's ability to detect gaps depends on where those gaps fall with respect to the phase of the stimulus rhythm and the entrained brain rhythm. This is just a single subject worth of data. We've now replicated this over, I would say 200 subjects, sometimes over multiple sessions. We've done many, many experiments like this. This is also reliable. So if you measure this for a participant and bring them back on a different day, their data look the same when they come back. So this is a reliable signature, a behavioral proxy of entrainment. These are just to show you a couple examples of other subjects. So here what I want to tell you is that behavioral patterns are consistent with entrainment of endogenous neural oscillations, so with entrainment of excitability fluctuations. So I'm going to switch now to the sort of meat of the talk And I'm going to talk about using non-invasive brain stimulation to study entrainment. So in my lab, we've been using transcranial alternating current stimulation, or TACS. TACS works by applying a weak alternating current, electrical current, to the scalp of a participant. And we know that this weak electrical current is capable of entraining endogenous oscillations. We know this from animal work, and we know this from work even stimulating groups of neurons that live in a dish. And depending on the electrode placement, sorry, this microphone is driving me insane. Depending on electrode placement, we can target specific brain regions. So you can see we have a specific electrode montage here for this participant, and this is an electric field simulation showing how much current is getting where in the brain. And here we're targeting primary auditory cortex. This was a target that we functionally defined based on an fMRI study that we did with the same subjects. So what's the advantage of using electrical stimulation? Why am I interested in this technique? We know that both auditory stimulation and electrical stimulation are capable of entraining neural oscillations. In addition, auditory stimulation evokes neural activity in response to sounds. And in any EEG, MEG signal, these signals are all mixed up. And as I said, no signal processing technique is going to take them apart. The important thing is that electrical stimulation does not evoke neural responses that weren't present to begin with. It's much too weak to do this. There's lots of work also showing that TACS can only interact with a brain state, a brain rhythm that's currently active, that's present. So this is satisfying the assumption that the oscillation already exists. We can't create a brain rhythm out of nowhere and interact with it. It has to be there first, and then the electrical stimulation is capable of picking it up. So we know we're interacting with endogenous brain activity, whether it's entrained or not. So we're using the same behavioral paradigm that I just described, so this gap detection and frequency modulation experiment. Here what we did was we have a sound rhythm, and with respect to the sound rhythm, we varied the lag, the temporal relationship of the electrical stimulation to take on one of 6 values, and I'll plot those here on a circle. So you can see the 0 phase lag condition is synchronized perfectly with the audio. The out of phase stimulation condition is anti-phase to the stimulus rhythm, and we've got conditions in between. We also included a sham condition, so this is where we turn the stimulator on for 30 seconds and then we turn it back off. So that initial ramp up is when people are most likely to feel that they've been stimulated and know that they're in a stimulation condition. After about 30 seconds, they don't feel anything anymore. So when we turn the stimulator back off, this gives us a baseline condition. And we did this in 2 sessions. We can only stimulate for 50 minutes per session. So we brought the participants back twice. So I'll show you both sessions of data. So here we wanted to understand, can TACS in principle interfere with oscillations that are already being entrained by a sound rhythm? And here we wanted to ask what the nature of that interference would be. And this has implications for how TACS might be used. There are a lot of ideas that brain stimulation might be used as an intervention, that maybe we could improve entrainment and improve listening success, especially in older adults or in noisy environments, for example. So we have to know how exactly we're interfering with those entrained oscillations. So we like to talk about 2 general families of hypotheses. These are not mutually exclusive. They're just our sort of 2 endpoints. And I'm going to describe those in terms of the behavioral dependent variables that we're going to be interested in. So we have 2 dependent variables. This is, again, the single participant gap detection hit rate data that I already showed you. We can quantify that in 2 ways. One is what I call behavioral modulation depth. That's how tall this modulation is. So for some participants, their behavior matters or depends a lot on the stimulus. And for other participants, their behavior doesn't depend very much on the stimulus. So that modulation depth, we think, is a behavioral proxy for the strength of neural entrainment. Another dependent measure is what I call preferred FM phase. So if we find the peak gap detection performance, what's the phase of the stimulus that yielded that? That's that person's favorite stimulus phase. That led to them doing the best in terms of the gap detection task. So the first family of hypotheses is that TACS might rewrite what the brain is naturally doing. So what I mean is that TACS might actually be capable of dragging the entrained neural oscillation into a new phase relationship with the stimulus. So when we shift the phase of the TACS relative to the audio, we'd be shifting the phase of the brain relative to the audio. So here I'm plotting cartoons of what that might look like. So in the 0 phase lag condition, the pink one, we expect alignment with the stimulus. This is hit rates, I should say. This is our behavioral proxy that I'm plotting a cartoon of. And as we dephase the electrical stimulation with respect to the audio, we're expecting to drag the brain into a new phase relationship, and that's going to change the preferred FM phase. So this hypothesis is tested based on looking at this preferred FM phase dependent measure. The other option is that TACS might interfere with brain activity. So the brain might keep its own natural phase relative to the audio stimulation, and in that case, dephasing the electrical stimulation relative to what the brain's doing would actually mean, think constructive versus destructive interference. So if we line them up together, we'd be able to boost the strength of entrainment, and if we cancel them out, if we use the electrical stimulation to cancel out the brain rhythm, we're going to reduce the entrainment strength. So when the TACS is aligned with the stimulus in the 0 phase lag condition, we're expecting an enhancement of that behavioral modulation depth. And as we dephase the electrical stimulation relative to the brain, we're expecting destructive interference. So this is a hypothesis that's tested based on this behavioral modulation depth parameter. These are just a few single participants, so you can see the type of data that we're working with. This is 3 participants in two sessions each. Gap detection depends on the phase of the stimulus into which the gap fell, so you can see these are fluctuating with the stimulus rhythm, as always. And you can already see, just by eyeball, that if there's any shift in the preferred phase of the stimulus, it's going to be really, really minor. So we will start there. I'll show you the phase data first. So I'm going to plot circle plots now. So each plot is going to be for one of the phase lag conditions. Color is session 1, gray is session 2, and then you have 2 resultant vectors, one in color, one in black for the 2 sessions. And remember, if the TACS is dragging the brain into a new phase relationship, we expect these resultant vectors to rotate their way around the circle, and that's absolutely not happening. So TACS is not capable of dragging the brain into a new temporal relationship with the sound, at least in this paradigm. So that doesn't mean that any entraining rhythm wouldn't be capable of being phase shifted, but here we've basically set up a competition between the audio entrainer, which is very, very strong in this situation, and an electrical entrainer, which is very, very weak. So it's not necessarily the case that in every experiment you're not going to see that phase shift, but we didn't see it here. But now we'll ask whether TACS is capable of interfering with entrainment. So we'll leave the brain in its natural phase relationship to the stimulus, and we'll expect to see constructive and destructive interference depending on where we're putting the electrical stimulation. So first, I'm plotting the data for the sham condition. So this is when we're not stimulating. This is our baseline. And this is behavioral modulation depth on the y-axis. So remember, that's the tallness of that behavioral function, behavioral modulation. And just a reminder that what we expect to see is a sinusoidal modulation of this dependent variable. So as we phase shift, we expect constructive versus destructive interference and in between. Ideally, with respect to the sham condition, we want to see a strengthening and a weakening of this behavioral modulation. I would point out that in the literature trying to use TACS to change listening behavior, it's very common to see that you can make performance worse, but it's very uncommon to see that you can actually make performance better. And that's really important to show if we're actually going to talk seriously about this being used in an intervention kind of capacity. So here are the empirical data. This is exactly what we see. So we see this sinusoidal modulation of behavioral modulation depth. And again, we did that in 2 sessions, so we internally replicate this effect. Here I'm just sort of brutally binarizing the data to show you that when we have, this is our sort of positive half of the cycle, and this is our sort of negative half of the cycle. And all I want you to notice is that we did significantly improve or strengthen, I should say, and weaken that behavioral proxy of entrainment. So here we found that TACS interferes with entrainment of neural oscillations by a constructive and destructive interference, at least in this case where we set up a competition between a strong auditory rhythm and a weak electrical rhythm. I'm going to talk about one more TACS study, but I'm going to pause real quick to talk about another property of oscillators. And this is preferred rate and the associated entrainment region. So preferred rate's called lots of different things, depending on the literature that you're reading. Preferred rate, preferred frequency, preferred period, natural frequency, eigenfrequency. You may have seen any of these names. The idea is that an oscillator has a preferred rate of oscillation. All oscillators do. So for our circadian rhythm, for example, the period's 24 hours. Oscillators have a bit of flexibility, so they can go faster and they can go slower. This is a necessity for entrainment. They have to be able to adjust to some extent. But they don't like getting too far out of their own comfort zone. So here the x-axis is the rate of a stimulus with respect to the preferred rate of an oscillator, which is right in the middle here. On the one side, the stimulus is too slow, and on the other side, the stimulus is too fast relative to the hypothetical oscillator. So what we predict is something like a tuning curve around the preferred rate. So we have entrainment strength on the y-axis here. When the stimulus rate matches the preferred rate of the oscillator is going to entrain very easily and with a lot of strength. The farther and farther we get away, the larger our detuning, you could say. The larger the difference between the preferred rate of the oscillator and the stimulus, the weaker entrainment's going to be. And the sort of width of this thing, the space in between here is our so-called entrainment region. So within this region, entrainment is possible. Outside of this region, entrainment is not possible. So an oscillator has to be flexible, definitionally, for entrainment to occur, but no oscillator can be infinitely so. So we're doing now the same task. It's a gap detection task in a noisy stimulus. But here the stimulus is not rhythmic. So we don't have an auditory rhythm. So we're not entraining anything. The electrical stimulation is our only entrainer. So the way that we set this up was instead of placing gaps into different phases of the stimulus, we placed gaps into different phases of the electrical stimulation. So here's a schematic of that. This is TACS phase now, electrical phase on the x-axis, and this is just a schematic of the electrical stimulation, the actual, the amplitude of that. We probed gap detection at 6 different gap locations. So again, if the electrical stimulation is entraining endogenous now, because they're not entrained to the audio stimulus, endogenous oscillations, then we expect to be modulating neural excitability, and we expect to see gap detection hit rates changing sinusoidally as a function of TACS phase. That's our prediction. We applied the electrical stimulation at 4 different rates, and we sort of roughly sampled the so-called delta band. And for each frequency, we're going to look at how strong this behavioral modulation is. In my lab, we're focused a lot on individual differences. So here we're not actually interested in differences between these stimulation rates. We didn't have any hypothesis that one rate was going to do a better job than another, but we were interested in whether different stimulus rates, different electrical stimulation rates worked better for different people. So the idea here is, or the question, I guess, is whether we can capture an individual's preferred rate of endogenous oscillations, and then use that to look for an entrainment region and test a critical hypothesis of oscillators. So remember this pattern. This is what we're predicting. And again, this is exactly what we found. So we have TACS phase on the x-axis and gap detection hit rates on the y-axis, and you can see that these middle points are better than these outside points, no matter what the stimulus rate is. So that's significant for all of the stimulation frequencies. And here again, I'm just going to brutally sort of binarize the data as an illustration. I'm going to group them into the best and worst phases, just to show you that there's, for each frequency, a significant difference. Hit rates are better in what we call good phases and worse in what we call bad phases, and the magnitude of that difference does not depend on the stimulation frequency. But again, that's not what we hypothesized. What we're interested in here is individual differences. So the idea is that one person might have really been strongly affected by this particular stimulation frequency, and another person maybe less so, but maybe a different stimulation frequency worked better to modulate their behavior. So as an initial analysis, so here on the y-axis, I'm going to put TACS effect magnitude. You can think of this as a kind of best minus worst. So just how strong, you can also think of it like the behavioral modulation depth parameter I already explained. How strong is that behavioral fluctuation? And this is a noise floor that we created using something like a permutation test. The details are not important. In that band or below means that the behavioral modulation was not greater than one would expect by chance. And these are individual data, individual participants in the dots, and then the summary is in the box plot. You can see that there are some individuals for each frequency who had a behavioral modulation that's greater than we would expect from chance. The interesting thing is that these are all different dudes for each frequency. So each of these groups these are not overlapping. So some dudes were very strongly modulated by the lowest frequency. It's different dudes that are very strongly modulated by the highest frequency. And so to try to summarize that, we submitted these data. So these, it's 4 data points per person, the TACS effect magnitude as a function of TACS frequency. We submitted those to a cluster analysis. And that cluster analysis revealed 3 patterns. So these are 3 groups of participants, 3 groups of dudes. So in our first cluster, we have a sort of, we have this group who were really strongly modulated by the slowest electrical stimulation, and progressively less so as the stimulation became higher frequency, as the rate sped up. In this third cluster, we have the opposite. So we have people that were very strongly modulated by the fastest electrical stimulation frequency. And in the middle, we have this sort of bandpass group that were most strongly modulated by the frequencies in the middle. And to try to interpret that, we implemented a computational oscillator model. So this is a Kuromato-like model, where we have a single oscillator that we assume different preferred rates for, and then we stimulate it with a fake TACS signal at all of these different frequencies. And by assuming an oscillator with a slow preferred rate, a medium preferred rate, or a fast preferred rate, we're able to exactly recreate, well, exactly. These look a lot nicer than our behavioral data, as always, they're model data. But we're able to qualitatively recapture these 3 patterns. So the low frequency likers, the middle frequency likers, and the high frequency likers. So the prediction of these 3 clusters actually comes out of an oscillator model if we give it different preferred rates. So different TACS frequencies entrained individuals' neural oscillations to different degrees, and behavioral patterns were predictable from an oscillator model for which we assumed different preferred rates. So what's next? Right now, we're in the process of re-recruiting participants from these 3 clusters. And we are testing them on an EEG version of the gap detection task where we use the rhythmic auditory stimulus at these 4 different rates. And so the question is whether using TACS we're actually identifying the preferred rate of an endogenous oscillation, and we're able to predict at what frequency they're going to best entrain to a driving stimulus in the world. So, to conclude the sort of empirical bit, non-human animal and human behavioral work can help us to separate neural entrainment from evoked activity. TACS interferes with neural entrainment via constructive and destructive interference. And TACS at different frequencies reveals preferred rates of endogenous oscillations in the auditory system. So the second point shows us the specific way in which we can expect for electrical stimulation to interact with oscillations and informs our designs going forward. And based on this third point, we think we can leverage this technique to test hypotheses of an entrainment, the presence of an entrainment region, which would be further strong evidence for entrainment of oscillation or of brain rhythms that are generated by an oscillator. I'm going to abuse the few minutes I have left to come back to this cartoon that I presented to you at the beginning of this very simplistic set of criteria for what it means for an oscillation to be entrained. So at the beginning I said the oscillation exists before the stimulus is present. Really what I mean is that the oscillation exists in principle. It can exist. It's not quite nuanced enough to say that it must exist. We must be able to see it in silence, for example. And without nuance, we might be hunting for something. We might be hunting for a prediction that, without justification, I guess. So an oscillator might be damped. That means without input, it decays to a fixed point. So when we stop stimulating it, the oscillation goes away. It follows from that that there's no oscillation before the stimulus starts. So if it decays back to a fixed point after the stimulation is over, before the stimulation starts, you wouldn't expect to see any oscillation. That doesn't mean that the activity generated during stimulation is not being generated by an oscillator. It just needs some external energy to produce its oscillation. So this might be problematic, for example, if we hunt endogenous activity during resting state, for example, if we want to know about the endogenous oscillations of the auditory system, silence might not be the best option. We might need to put a little noise into the system, for example. The auditory system it's not naturally very peaky in terms of a frequency domain representation. It's not very easy to see anything that looks like an endogenous oscillation during silence, but actually the entire spectral profile of what the auditory system is producing changes as soon as you give auditory input. So as soon as you give a bit of noise, for example. We also tend to assume in some of the literature that in order for entrainment to music, to the beat of music to be a thing, that we need to see this, endogenous 2 Hertz oscillation in the auditory system, and that must exist on its own. And again, what I'm trying to emphasize is that we might not expect to see this depending on the situation in which we're hunting. This also has implications for how we use electrical stimulation. So I said already that TACS can only interfere with active existing oscillations, which is why in the experiment I explained to you, we're stimulating with noise, even when we didn't want to use a rhythm. We're trying to activate those endogenous auditory oscillations so that we can actually talk with them. The second part, I think, is a bit less controversial. So the oscillation adjusts. That has to be true, but it's not infinitely flexible. I think that this has implications for randomly choosing a stimulation frequency in a one-size-fits-all way for all of our participants and expecting to see entrainment to the same degree. We've already talked about this, but the last study that I explained, what I'm trying to emphasize is that preferred rates and entrainment regions might not look the same for everyone. And so in my lab, we've been working on techniques to try to estimate these preferences a priori and then target each person's preferred rate and look for their own personal entrainment region. The last is my pet peeve assumption. So that's that the oscillation continues. There are three possibilities for how this might happen. The one I see in the literature most often is that the oscillation continues with the same properties as the stimulus that was just being presented. So we tend to look for self-sustained oscillations in silence after the stimulus has ended as evidence for true entrainment. But there are two other possibilities. One I've already explained. That's the third one. I'll say that next. The first, which I'm showing you here, is that the oscillation continues, but it decays back to its own preferred rate. And you saw this in Edge's talk this morning. Another possibility is what I just explained a few minutes ago, that the oscillation is damped, and it decays to a fixed point, so it decays to no oscillation. And in reality, these latter two options are metabolically less expensive, and so theoretically more likely to be what's happening. So the oscillation, I would say, can't be expected to just keep on going forever with the temporal properties of the auditory stimulus unless it has a reason to. So now we're back to talking about creative experimental design. If we give the oscillator a reason to try to predict something that's coming up in the future, for example, we might expect the system to internally generate, we might say top down, we might say attention, but internally generate some energy to keep that oscillation going rather than decaying in either of these more natural ways. And I'm happy to discuss this more, and I'm sure we'll talk about that in the panel tomorrow. So I'll go back to my empirical conclusions to end so I don't leave you on that sort of, I don't think it's pessimistic, I think it's realistic, so I don't leave you on that theoretical note, but I'll go back to my empirical conclusions, and thank you for your attention, and I think I have time for a few questions, I hope.
Thank you so much, Molly. In the great words of a wonderful neuroscientist, Molly Henry was such a killer choice, dude. This talk has been awesome. And that's come in all the way from Montreal from Connor. So thanks, Connor Speech, for your input. I think we already have a first question.
Okay,
so Peter.
let's do it.
Thank you very much, Molly. That was really impressive. And it makes me wonder, so you were focusing on the auditory system.
Yeah.
So auditory cortex, trying to manipulate and measure what's going on there. But I think some of the issues that you touch on, they raise questions about connections with the motor system.
Oh, absolutely.
So if you're talking about preferred tempo, there are ideas that this is somehow related to the motor system. It influences, and motor capacities might do that. And also the idea of some self-sustained things. You could imagine a loop going on where it kind of reinforces one another. So I'm wondering if that sort of fits into your big picture, and if so, kind of how, and if there's evidence for it in the line of work that you're doing.
Yeah, it 100% fits with what I'm talking about here. So here today, I've concentrated on the auditory system. We also do some work in the lab bringing our bodies into the picture. When I talk about oscillators and when I talk about preferred rates and entrainment regions and things like this, I'm not thinking of I don't actually believe that any part of the brain or the brain itself is an oscillator, but this is a sort of a way to describe. And what I think is that the oscillator that we're trying to characterize depends on the system that's in play. So here we're talking about auditory perception, and the stimuli that I'm using are not even beat-based. We don't have to internally generate anything. But when we bring our motor system into play, or we start using musical rhythms, for example, where we might have to impose a structure, we're talking about a different system. And some of the work from my group has shown that preferred rates differ. We can't predict preferred rates for all things from all other things. So there are some relationships between preferred rates for perception and production, for example, but bringing the body in tends to slow preferred rates down, at least in what we've seen. But the task has to be really similar. So if you then move to try to predict preferred rates for listening to music or tapping to music versus a spontaneous motor tempo task or a sensory motor synchronization to a metronome, for example, these systems actually tend to have different preferences. They have different preferred rates, and I would predict they have different entrainment regions. We also think about the width of that in terms of adaptability and how flexible a person is. So I think it's 100% in line to say, you know, now we're talking about a system that's a network and incorporates connections between the auditory and motor system, the brain and the body. Yeah, same principles apply, but I truly believe that we have to characterize those systems differently because it's a different network now, and we can try to relate them to each other and see what kinds of predictions we can make, you know, across these systems, but we sort of have to look at it with fresh eyes, I would say. Does that answer your question?
Yes, it does. Thank
Okay.
you.
Thank you so much. We'll try to balance the in-person questions with the online. We have one in from Connor, actually. So any plans to correlate these endogenous neural and behavioral oscillators with participants' spontaneous motor tempi?
We actually have collected spontaneous motor tempo data for every person, for every experiment we've done in the last 5 years. We also tend to re-invite participants over and over again, so we can look at their preferred rates across different paradigms and in neural stuff versus behavioral stuff, for example. So we have, we have spontaneous motor tempo for everything we've ever done. It's not a great predictor of very many preferred rates. It predicts preferred rates for tapping to metronomes super well. Again, I think this is because of the system that we're talking about. You know, spontaneous motor tempo, you just tap your finger at a comfortable rate. That predicts how you like to tap your finger to a metronome. It does not predict how you like to listen to music, what kind of frequency modulation you like to listen to, what endogenous frequency we're able to pick up using TACS. Those things are not related to spontaneous motor tempo in our hands. But again, the tasks have to look like each other because the system needs to be similar. You know, so what's the neural network underlying what you're doing? What's the body contribution to what we're doing? What are the cognitive demands for what we're doing? That all changes the system, and the farther away you get between systems, the less likely they're going to predict each other. And so spontaneous motor tempo it does work in some cases, but it's really specific, actually.
Do you then have like a hunch of where these preferred alternating current rhythms then are coming from within the participants you've had?
Those, I believe, are endogenous, sort of preferred rates of endogenous activity that the auditory system would spontaneously generate when you're listening to sounds. It doesn't have to be a rhythmic sound, but when the auditory system's being driven in some way, when there's energy being put in from the environment to the auditory system. I have, you know, speculate like crazy, but every single person's auditory system looks different. So we have different gray matter concentrations, we have different connectivity strengths between the auditory system and different parts of the brain in terms of, you know, white matter tractography. There's different amounts of myelination, there's different cortical folding patterns for each person. Like, no two people's auditory systems look the same. And so, I mean, a dream scenario would be to actually understand why there are differences. I mean, I think it's clear that there are differences between people, but where those come from in a sort of structural or like a real sort of brain way, I don't know.
Mm-hmm. But I mean, even if we don't know where they're coming from, it sounds like, I mean, this reminds me of personalized medicine with
Exactly.
the examples you gave in applications. That's really cool. Are there any questions from the audience again? Here you go.
Thank you very much. You were hinting or saying that it's not reasonable to think that the oscillator would continue spontaneously unless there is a good reason. Can you elaborate on that? I was thinking maybe a gap in the stimulus and then return to the stimulus would drive the system to continue, but maybe you can elaborate on what you were meaning.
That's exactly what I mean. I think if we're stimulating and then we stop the stimulation and the participant, whether it's a human or a non-human animal, knows, okay, sequence is over. I can quit doing my task now. There's absolutely no reason that we would assume that would keep going. I mean, it might in the case of, you know, in the visual system, alpha oscillations are very much more measurable during silence and measurable after a stimulus. It still decays back to its own preferred rate, actually, but Yeah, anyway, yeah, so to answer your question more precisely, that's exactly what I think. So if we would create a situation where you have a rhythm and then there's a break and then you have to do a task. I mean, some of the tasks we've seen presented today already would be good candidates for something like this, where you have a break that you have to extrapolate through. That's actually the task. And then I saw a recent talk from Fleur Bauer, who did something like this, where there was a sequence and then a break, and then you have to judge something about a tone that happens in the future. And there they actually did see a peak at the stimulation rate during the silent bit, because you have to operate on something that's coming up and you have to predict the timing of that thing. So yeah, I think it's all about experimental design, actually, to be able to see that.
I'm so sorry to cut this question answer period short, but we have only a 10-minute break until the next presentations,
I
and
don't
I
wanna cut
will
into that.
send Emily Graber and Roger Moore's questions to you over email and connect you with them, if that's okay. Thank you so much. Another round of applause for Molly, please.
Thank you.